Welcome to Ajad Bioacademy. Today I will talk about receptor. Firstly, receptor. Firstly, you have to know something, then we'll go into the details. The receptor is, in generally, which substances receive something that can be easily called receptor. But in pharmacological perspective or in biochemistry perspective, receptor definition will be somewhat different. That is, it will receive the ligand and that ligand will make changes of that receptor, and that receptor has two basic two sides. One is binding domain, another one is response domain. Okay, and it will change its conformation, and that will just help to give biological response, or it will instigate the signaling pathway, or you can say it will help the cellular response. Okay, so this is receptor. So what would be the nature of the receptor? Basically, receptor nature is protein in nature. This is macromolecule. But what type of proteins? Is pure protein or some additional substances are attached with it? Basically, it is glycoprotein in nature. Basically, okay, mostly, but not all the receptors are glycoprotein in nature. When carbohydrate is glycan part is attached, so it is a glycoprotein. Sometimes lipid attached there, sometimes phosphate attached. So it can be different structural pattern. So receptor is a macromolecules, protein in nature substances, which is basically present in the membrane, also present within the cell, and sometimes it also be present in outside of the cell. Okay, so roughly I will try to give you some initial idea, and then I will go into the details. Okay, so firstly I wanted to say about the transmembrane receptor. Transmembrane receptor, that is which is present in the membrane, like a crossing, one part is outside of the cell, another part is inside the cell. And the second one is intracellular. Okay, intracellular receptor. That means within the nucleus, here, cytoplasm, or within the nucleus. So you can say intracellular. Separately, you can say this is the cytoplasmic receptor, or here you can say this is nuclear receptor. Okay, and third one you can say extracellular receptor. That means outside of the cell, extracellular receptor. Okay, so I already said our discussion matter is receptor based on the pharmacological perspective. So, drug or ligand can show its activity in different way. That can bind with the receptor, that can bind with the enzyme, that can bind directly to the effector substances, or it can change the physiological condition, or it can work on the ion channels. In different way drug can work. But our today's discussion topic is receptor. So why receptor is necessary? Drug will act, but why receptor is necessary? And where it is located, already I said. But how it will work? Roughly, I will give you, and then when I will try to discuss all the receptors one after another, then I will go into the details. So firstly, if you think this is a lock, okay, and you can say that this is a key. Another things, I will give you an example. Just I'm considering transmembrane receptor to give you some basic idea about receptor. So this is the membrane, and this is a receptor. Okay. So it must have two domain mainly. It must have two domain, and another several domains may have within the receptor based on the types of the receptor. Suppose if you think your nuclear receptor, that is glucocorticoid receptor, it has multiple domain. Okay, but if you consider as a receptor, it must have two domain. That is ligand binding domain, and it's the signaling domain. That is your response domain. That means receptor is protein in nature, so it is made up of amino acid chain. So it must have N-terminal and C-terminal. Okay, so N-terminal domain or C-terminal domain. Here the binding domain. So you can say it must have a binding pocket. That pocket may be large, that pocket may be narrow, that pocket may be open. Okay, because it's a protein in nature. So protein is the receptor protein is nature is folded. So different side amino acid has different nature. Someone is just carrying aromatic ring, someone is carrying positive charge, someone is carrying negative charge. So it just depends on the nature of the protein or nature of the amino acid side chain. Its role is different, but two things is very fixed. One is the binding domain and is signaling domain or response domain. So the binding domain, it has some nature to bind with the ligand, and then the ligand must have some ability to work on it. So how it will work, I will just give you with the example that and that. Okay, so here this one, you can say this one is the ligand, this one is ligand, and it will bind with the receptor. How it will bind? It is a ligand, then it must have functional group or a specific structure. And the receptor is made up of amino acid. So amino acid has different types of amino acid, and it has different role. So according to their nature and their binding domain pocket, it will fix with it and it will make a bond. Okay, it will make a bond. The bond forming capability depends on the nature of functional group, electron proton exchange. Okay, so different types of bond will form here according to the nature of the ligand and the types of receptor. That can be your hydrogen bond, that can be van der Waals force, that can be even ionic bond and even covalent bond. Different types of bond can form. So bond forming ability of that ligand to the receptor, that is called the attraction, that is affinity. Okay, so this is called affinity. So ligand must have affinity to the receptor, and when they will meet the criteria to bond, to make a specific bond, then it will just bind with a specific binding pocket of the receptor. Okay, so some substances, the ligand which has affinity, that is attraction towards the receptor, and that has capability to form a bond, that is called affinity. That is attraction to the receptor, that is called affinity. So basically the ligand, you can say, suppose you can consider to understand easily, that is agonist. Okay, so agonist must have a affinity with the receptor. Okay, what will happen if it is considered as an agonist? So it should have another function, that is, it will make the changes of the conformation of that receptor, because this ligand is not allowed within the cell because its molecular size is larger and it's hydrophilic, so it's not allowed within the membrane. So it will just give the message through the receptor. Okay, so how it will give message? So that ligand will make changes the conformation of the receptor. How it will change the conformation? That is ability. Suppose if I want to break down a table, so what should I do? At first I have to grab the table, and then I have to just apply my intrinsic ability, my force, energy. That's the specific ability to change the structure of the table. Okay, so such type of ability, own ability, intrinsic ability, must have of the agonist. So that agonist will make changes the conformation. How it will change the conformation? Because the binding frequency is one of the major things. It will just try to locate the specific site, and several times it will just attach and detach, and then finally it will change the structure. That is binding affinity. That is agonist must have higher binding frequency. Okay, and another one is that is, it will, the receptor has a specific structural stability because it's a folded protein, a receptor protein. So it's a internal different bonds and different structural pattern. So it must, the agonist must just change the overall structure. That means its conformation to activate the response domain. Okay, so what substances will depends on it? That would be the nature of the amino acid, that would be the nature of the drug. So sorry, that is the ability of agonist. So everything depends on the agonist nature. So how it will make changes? Its inner ability to change the conformation of the structure of the receptor. Okay, so that ability is called efficacy. Okay, efficacy means the ability of the drug to change the conformation of the receptor. That means ability of the drug to activate the receptor. When the conformation will change, then the response terminal will be activated, triggered, and this terminal is attached with different types of internal protein or even channels or something like that. Okay, so signaling pathway will open. So the intrinsic ability of the ligand is called efficacy, and its binding attraction or binding to form a specific bond with the receptor, this is called affinity. Okay, so how we will connect with that lock and key mechanism? That means if you want to open the lock, you must put the key to the specific port. Okay, that means a specific binding site. When you will just insert the key into the lock, then what will happen? You must press or you must push, or you can just twist to open the lock. But just you will push it and you will just twist and then lock will open. No, you have to press, you have to apply some energy, because the lock has integral internal barrier potential. That is, it has stability. If you want to change the internal structure, then you must put the energy, or the lock, sorry, that key should have a ability to change. Okay, so like that, the receptor has a structural stability, structural pattern. So if you want to change this, you must have ability. So the key is just as like as the structure is as like as the port of the clock, as like the binding domain of the receptor, and the structure of agonist, structure of ligand, it must be similar. Okay, like that, lock and key. And then the ligand, that is agonist, it must have ability to change the structure as like the lock and key. Okay, so when the structural pattern will change, then is conformation will change, and finally internal signaling sequence will be activated. Okay, so this is the actually the receptor pattern, receptor structure. Okay, any type of receptor, it must have two things: one is binding pocket or binding domain, and finally response domain. And another domain, multiple domain may have that they have different function. Suppose if you think the nuclear domain, it must have a DNA binding domain. Okay, so like that. So here the receptor is, it will receive the drug, and then the drug will apply its ability to the towards on the receptor, and that receptor will be activated, and the activated receptor will activate the internal response. So if you consider this macromolecule as a receptor, what would be the criteria? It's the macromolecule here, and it's the protein in nature. Okay, and it will bind with the agonist or ligand, and that will change the conformation, and then that will gives a biological response, or you can say a cellular response, or you can say activate the signal sequence. This is the receptor. Okay, so if the ligand has a ability to change the structure of the receptor, that is the agonist, or if something like just similar structure like that, and it has affinity toward the receptor but has no ability to change the structure, so this is called antagonist. That is, you can consider like the chairman should have a chair to sit on it, and it will walk the official walk, but a peon has very attraction towards the chair to sit. But if you just allow him to sit on the chair of the chairman, then peon can be able to function? No. So it will just has affinity towards the receptor, but it has no ability to change the conformation. So it has affinity but has no efficacy. So this is the antagonist. Okay. So this is not my concern today, but I will just try to give some just idea. Okay. So I will just go on after another, the transmembrane receptor, intracellular receptor, and extracellular receptor. Okay. So now come to the transmembrane receptor. Transmembrane receptor. Transmembrane receptor. So transmembrane receptor, already I said, this location is the within the membrane. Okay, so like that. And what types of transmembrane receptors are there? That means, it will just cross the membrane. That is transmembrane receptor. And the drugs which actually give its function on the basis of receptor binding, it is the most of the So firstly, the transmembrane receptor. Okay, so already I said this is the membrane. This is present on the membrane as a transform. Okay, it's crossing the membrane. So what are the types of transmembrane receptor? It will only just cross several times, or one time, or multiple times, as per the basis of the pass of the membrane. It has several types. So come to the point. First of all, one is one pass receptor or single pass receptor. Okay, like if you think this is a membrane, and it will pass one time. Okay, it will pass one time. So this is one pass receptor. This is the binding domain. Here ligand will bind, and this is the cross membrane sequence, and this is, you can consider as a response signal or C terminal. So that is the receptor. It's one time cross the membrane. So you can say this is one pass receptor. You can say single pass receptor. Okay. And the example of that type of receptor is, you can consider easily insulin receptor. Okay. The insulin receptor, and you can also consider as a tyrosine kinase receptor. This is a single pass receptor. And second one, if you think, that is 4 or 5 times it will cross the membrane. And if you just consider, this is like that receptor, It has like that, and it has the binding domain. It must have a binding pocket, binding site. If you consider this one, and this is the channel, and this channel, through this channel, ion will just enter. So this is ion. Okay, so ion channel you can consider, and it is depend on the ligand. So ligand binding channel, that is, you can think ligand gated ion channel receptor. Okay, and that type of receptor is basically, you can just draw it that way. It will just cross 1, 2, 3, 4, or 5. Okay, 4 or 5 times it can cross the membrane. So you can consider as a 4, 5 pass receptor. Okay, and the example is ligand gated ion channels receptor. You can consider as a GABA receptor. You can consider as a nicotinic acetylcholine receptor, like that. That means ion channel, ligand gated ion channel receptors are types 4 to 5 times time pass or cross the membrane. That is the 4-5 pass receptor. Another very common receptor, that is, it will pass 7 times. 1, 2, 3, 4, 5, 6, 7, like that. Okay, and this is called, here you can say ligand, and this is your response site, and this is the crossing part of the receptor, and this is called 7-pass receptor. You can easily say 7-pass receptor, and it looks like a snake, so it's a serpentine receptor. Or you can consider example, you can think this is G protein couple receptor. Another name is metabotropic receptor. So serpentine receptor or 7-pass receptor. Okay, so we got 3 types of transmembrane receptor. Another special type of receptor is there, and this is, you can think this is your presynaptic terminal, and this is post, and it has some receptor in post terminal, and also in presynaptic terminal has some receptor. So if you think this is the presynaptic vesicle, and when it will just reaches, this is the synaptic cleft, and this is neurotransmitter. Okay, and it will bind with the receptor and it will work. This is very common phenomenon. But sometimes some neurotransmitter will go back. Okay, it will just go back. So that type of receptor is 1, 2, 3, 4, 5, 6, 7, 8, 9, 12 times it will cross, or it will pass the membrane. Okay, and this receptor, you can think reuptake, monoaminergic receptor, monoaminergic neurotransmitter receptor. Its function is to reuptake the neurotransmitter to the vesicle, presynaptic terminal. Okay, and that receptor, you can say serotonin reuptake receptor, you can say, already I said monoaminergic, that's catecholamine neurotransmitter. That is, you can also consider dopamine, you can consider noradrenaline reuptake receptors, and this is types of 12 times it can pass, it can cross the membrane. So it is called 12-pass receptor. Okay, so the transmembrane receptors basically single pass receptor, that is insulin example, and ion channel receptors, GABA or nicotinic acetylcholine receptor, and 7-pass receptor, G protein coupled receptor, that can be adrenergic alpha beta receptors, and this is you can say serotonin reuptake receptor, you can say dopamine reuptake receptor, like that. Okay, so this is the transmembrane receptor. And now I will move on the intracellular receptor. Intracellular receptor. So if you think this is your cell, and this is your nucleus, and this is your genetic materials, here. Okay, so the receptor will be within the cell. So suppose if you think this is your one receptor like that, okay, present inside the cytoplasm, and another receptor is present within the nucleus. So where it is present? It is present within the cell. So combinedly you can say this is intracellular receptor, as I said shortly, intracellular receptor. Okay, so precisely this one is present within the cytosol, that is cytoplasm. So you can say this is the cytoplasmic receptor. Cytoplasmic receptor. And here the receptor is present within the nucleus, nucleosome. So this one is nuclear receptor. Okay, so both are combinedly called intracellular receptor. Okay, and some ligand will bind here and it will change the conformation, and of course it is binding domain and also has some another domain, DNA binding domain, because ultimate goal is to reach to the nucleus. Okay, this cytoplasmic receptor. And so you can say this both you can say the nuclear receptor, because its receptor ultimate goal is to work on the nucleus. Okay, so if I just give an example, you can say cortisol, glucocorticoids, you can say cortisol or glucocorticoids, another you can say estrogen, you can say progesterone. These steroidal hormones can basically work on the cytoplasmic receptor. Okay, and some nuclear receptor that basically you can say T3, that is triiodothyronine, active form of thyroxine, and you can say vitamin D. Vitamin D can work on the nuclear receptor. Okay, and how it will work and how the cytoplasmic receptor reach to the nucleus, I will just explain in later one. Okay, in separate lecture. Okay, so the third one I will just explain shortly. That is extracellular receptor. Actually, extracellular receptor is not considered as a mainstream receptor, but sometimes it works like a receptor. Actually, receptor because it meets the criteria of the receptor, because it should have a binding domain and also should have a response giving domain. Okay, and it will change the biological function, or it will give the cellular function. So it is as it is present outside of the cell, so it has a biological function. Okay, that means after binding to the ligand, it will change the biological function. So that is the criteria of receptor. Okay, so extracellular. So that receptor you can consider as a cytokine receptor, that can interleukin receptor, that can be CX2 or so on. So that can be receptor of IL-6, and that work on IL-6 receptor. Okay, you can think of antibody. Nowadays, it's a very good, promising area to develop drug, because antibody-oriented different types of disease, and some drugs are targeted on the antibody. So antibody, the drug will bind to the antibody and it will change the antibody function. So that is the receptor. Antibody itself can be a receptor. So that is the extracellular receptor. If you think another one, that is antithrombin 3 receptor. Antithrombin 3 receptor. Antithrombin 3 receptor, and the heparin will just work on it. So it's also extracellular receptor. If you consider on different types of substances like vascular endothelial growth factor receptor, this is extracellular receptor, and it has several receptors. Okay, so extracellular receptor means it is not present on the membrane or transmembrane or within the membrane. It's present outside of the cell, and solely it will help to function, to show the function of the ligand, and ligand will bind with it. Okay, So it will inhibit or it will activate the biological function that will depend on the ligand. Okay, so these are the extracellular receptors. So I will just quickly I'll try to sum up. Receptors is a macromolecules protein in nature substance which is basically present on the membrane or also present in the inside of the cell, and based on the ligand nature it will work on the different types of receptors. Sometimes receptors present an extracellular region. Okay, so based on that receptors are three types. One is transmembrane receptor, intercellular receptor, and extracellular receptor. Okay, and receptor uh, has ability to bind with the ligand. That means ligand and receptor has a binding capacity because the structural similarities. Because receptor will carry the pocket binding pocket, and the drug must have a functional group or structural criteria that will make a bond with the receptor. That is called affinity. That means attraction to the ligand with the receptor. That is called affinity. Okay, and after showing its affinity, that is binding, form a bond, then it must show the activation of the receptor. That means it will activate the receptor. How it will activate? It will make the changes of the conformation of the receptor. As the binding domain already on the binding domain, the ligand will bind, and inside the cell, it has the response domain. So different types of intercellular substances or some substances will attach with that and it will be activated. Okay, that will work on it. So different types of receptor already I tried to cover with it. So the ability of the ligand is the intrinsic ability that is called efficacy, and it will just it has a higher binding frequency to the receptor, and it will change the conformation by its own ability. That is called efficacy. Okay, so this is all about receptor. Thank you very much.